Hello, welcome to this webinar. And the first part, I'm going to comment on the calculation of the half-life, followed by an analytical treatment of a simple gene expression regulation model. And finally, we have a look at the toggle switch, a model that shows how to create bistability using simple assumptions about gene expression, which has application in synthetic biology. And here you can see equations that have to do with the calculation of the half-life. So I'm not going to go through all of these steps. You just can watch it for yourself, quietly read through it. Just to comment on it, we have an equation that would be the result of the mathematical analysis of a model. So that's not the model itself, but it's actually the result of solving the model, the solution. And that solution, in this case, is a function, is an exponential function. And because you can see it's exponent of minus ct, so it's a decaying exponential function, exponential decay. And using only two parameters, namely y naught and c, knowing those two numbers, y naught is the initial population. So what population do you start with? If you model population decay, then it's the initial population, y naught, that you can obviously adjust to your experimental or observational condition. And then the c is a parameter that allows you to regulate how steeply that decay goes down. So if you have some data, you can actually use y naught and c to fit your function to the data. And then in order to find out what is the half-life of that fitted curve, you can do a couple of rearrangements. And the important thing of which is that first step. I'm not explaining the other ones, but that first step, namely, what does it mean when you're looking for half-life? What you're looking for is the point in time when the actual concentration, so y on the left-hand side, is equal to half the starting population, right? Half-life referring to how long does it take to get to half of the initial value. So what you would do is you write this condition in a mathematical way, which is the population y is equal to initial population is y naught divided by 2 is half of the population. And then you use this expression to put in y naught on the left-hand side of this equation. Of course, y naught divided by 2, that's how it is. And then you have an equation that you then can go and try to solve in the end for that half-life. Half-life is how long does it take to get there, right? I know where do I want to get. I want to get to half the population, which is y naught over 2. But how long does it take to get there? So how long will it take until the function has reached that point of half a population? And you then rearrange this equation a little bit and you find out the time, then the way, and it shows it depends exclusively on that parameter c, on the speed with which this function decays. So if c is small, then it's a slow decay. If c is large, it's a fast decay, and so on. So that adjustment, so that you can fit to an experimental curve, that gives you the half-life. And you can, after fitting, if you have that achieved through fitting, you can then directly read out the half-life as, in this case, the logarithm of 2 divided by that constant c. So from a mathematical point of view, there's nothing very fancy, just rearranging things from the left-hand side to the right-hand side of the equal sign. And of course, you need to know, you need to have the knowledge, and you can get this in our mathematical background, that in order to get rid of an exponential, you use the logarithm. And that's how the logarithm enters in this equation, because you have two sides of an equation. Both sides you use to apply the logarithm to, and then you get the logarithm of half or minus the logarithm of 2 on one hand side. On the other hand, you get rid of the exponential, which is convenient because that allows you to solve this equation for t, which under the special condition that we have defined at the top happens to be the half-life. Right, so that was the introduction on the mathematical side. And let me just bring up in the background my code so I can have a look at this. This is still here, so I can take this out of the place for the moment. And what we're going to do is we're going to open a file in MATLAB, and I have this prepared. And I took this from that script actually, just as a template. And the template contains the assignment task. So we can go along and see how would you go about solving this problem that is considered here. Solve the problem and solve the equation, solve the differential equation, and give me a couple of facts about it. So what does it say? The differential equation is given. An interesting thing is here that unlike the model that, unlike the example that we just discussed, two differential equations, one for a first population X, a second population Y. And if we talk about gene expression, then it would be two proteins that have an interaction via one of the proteins being a transcription factor that has an impact on transcription or creation synthesis of the other. So that's the background. You need to refer to the materials to find out how that actually works. And what we're going to do is we're going to look at this equation and try to find a solution and trying to plot a function of that solution from that equation, which is the actual model, right? So remember what we looked at in this first example was the solution already. If we have a differential equation model where we say, how is the interaction going to go on, then we need to find out the solution first. And once we have the solution, we can then plot the graph and look at exponential decay, rise, or whatever we have. Right, so if I just take that and assuming that you also have been able to access this, so either you know the assignment or you have downloaded this or you can somehow follow me with what I'm doing, then the first thing what we want to do is we want to solve this equation using a function called dsolve. So this is a symbolic solution. So dsolve will be solution of a set of differential equations. So it can use multiple differential equations. We need that because we have two. And it will give you the solution directly if you apply it to the differential equation. And as we will see, you can specify certain conditions under which you want to solve them. So let's start with this. Let's try sims. So our symbolic variables are, and this is now the important way, we need to specify the variables and we say what do they depend on. If we use t, then we'll specify that they are functions of time, so concentrations that change over time, and that will be our aim to recreate this. And if we have two, we'll have x of t and y of t, just put it in like this. And in that model that we have up here, we have parameters g, b, and c. So I allow for symbolic parameters g, b, and c. In addition, there is a specification, consider a process with certain values for those parameters, C, G, and B. They have certain values specified here, but also initial concentrations. So like we had this Y naught that we had in the derivation before, now we have X naught and Y naught as initial concentrations of the two proteins that we are interested in. So we're modeling protein concentrations, and remember X was the protein of interest, the one that we are interested in its expression, and Y was a different protein that has regulatory properties that modulates the output, which is X. So, and then we have a specification, namely the initial concentrations are supposed to follow these numbers. I don't know where they come from. So just initially the protein of interest is in a very small concentration present, but you have a large amount of the regulatory protein. And so what will the interaction cause? What type of temporal profile will the interaction between the two cause? So the equations are given here. So let's just start by writing the equations in the correct way such that we can solve them directly. So we call them equants. We assign a variable to this called equations. And because we have more than one, we put them in square brackets. So it's an array of first equation and second equation. And now we need to put in the equation. The differential equations, so we put them in as x comma t. That was a lowercase t, so I think it needs to be case sensitive. And I need to specify the x of t, but I also need to specify the y of t, both of which are differentials. So diff of x of t, so the differential of x of t and the differential of y of t, so that temporal change, that's what it means. And now I need to say, what are those changes? So it's important to remember that this equal here is a double equal, a Boolean equal, so to speak, which is the convention for this solver to operate. So, and if I look at the equation, it's gy minus bx. So we need to put that in as g times y minus b times x is minus, not equal, minus b times x is the equation for our first variable, which is called x. And similarly, the equation for y has this equal equal, and what was it? Very simple. It was minus, no, it was just c, not minus, it was just c. So we can do that. We can write in c. That's simple enough. And later we can modify that and will be changed. But let's just start with this one here, c. And so now we have the equation specified fully. We can put a semicolon because that's no output that we want to consider. So we have the equations. And second, we define the conditions, and we know what the conditions are because it says very explicitly, take certain types of initial conditions and put them in directly. So we have, of course, having two variables, we have two initial conditions. We can specify them as x naught. That's x at the time 0 equal to x naught. But if I say x naught, then this is something that I need to identify. I need to create a symbolic variable x naught, and then I can set it to this, so sort of just as a symbolic variable, which later can take a number. And then we have the second thing, which is just similar in structure, y at time 0 equal y naught. And if we call it y naught, then we have to specify up here y naught as a symbolic variable as well. And next thing would be then to solve, to get a solution of this equation. And the function that we're going to use is called dsolve. So the symbolic solution for differential equations, dsolve, differential equation solve, so to speak. And we just have to specify what equations and under which conditions. So it goes around parentheses. EQNS was the name of the equations. So I put that in. And I specify my conditions, which I called cont. And that should give me a solution. Let's just see if something happens. And what we can see, if I just execute the code, see that the initial conditions have been specified. The solution to this is a structure, struct. It's a structure. It's contained in this variable s now with two variables y and x specified, and there's some information about what that field looks like. So if we now, instead of just wanting to know that we found a solution, we want to actually see the solution, This is very easy to do. Namely, we can just get this out and use a trick. Namely, one thing that we have actually done before, use the simplify and the x solution. So the solution for the first variable, we can access via s, the solution dot x. If you remember, it was y and x that we have available. So s dot x will be the solution for the first equation, and very similar in structure. We can get y, and we do again simplify s dot y. Close the parentheses, and we don't put any semicolons, so the solution should just be coming up down here. And we can see the solution is MATLAB did something, some funny calculation, and rather than being an exponential to the power of minus ct as we had before, now it looks a little bit similar in the beginning, exponential minus bt in this case, but oh my goodness, then so many other things, a very long expression that you get, and something that you definitely don't want to do by hand anymore. That's the solution for the x variable. So the first solution and the second solution for the second variable is much simpler. Namely, it's just y naught. Starting point is a constant value plus c times t. So just as t increases, as time goes on, it will just grow with a speed that is regulated by a parameter called c in this case, because t is multiplied with c. So it will give us the speed with which that y variable grows. And of course, then what is about the initial condition? Y naught is here, but to know the solution for the first variable, we also need to know the initial condition, not only of the first variable, but also the initial condition of the second variable. So these are actually connected. Solution for the first variable contains information about both species and incidentally contains all three parameters c, g, and b. Okay, so we have solutions x and y. Yeah, so what I have is now the variable x contains the solution and y contains the solution. So I just clear my screen here, but we have them in variables, so we can deal with them. Right, so let's see. Let's go and get this into a MATLAB function that we can plot. So we need to convert this symbolic function that we've called capital X or called capital Y, and we want to convert that into a MATLAB function that we can actually use to plot. For abbreviation, we don't want to do that in detail for the both subpoints. I leave that for you. I think you can do it. What we can do is we can modify the code and go to this question directly, modify the MATLAB code to solve the differential equation such that the equation for y is equal to minus c times y. So it's a bit more complicated. And with the same initial condition, so which is actually easy to do because the only thing that we need to change is the differential equation. So we go back into our code and we say c times y, just checking, minus c times y. So we need to put that minus sign as well. So we have now a new differential equation, and if we execute that code, we'll get a solution that is shown here in the command window that looks different for both x and y, but notably in y looks very similar to something that we have already seen today. And can you recognize y is equal y naught times exponent of minus ct? It's just a thing that we had initially for the calculation of the half-life. Right, so let's go and take this solution for this modified gene expression system, and let's have that plotted. So what I'll do is I make a section, new cell here with the double percentage sign, and let's have a look. Let's have a look at first the concentration of y, because that looks so simple. So I have a MATLAB function y, an algebraic version of y by using this MATLAB function. Using MATLAB function convert that solution y, that capital Y, to an algebraic function. It's a symbolic variable. We need to convert that to an algebraic variable. So let's do that. Do you remember how to do that? We specify the vars argument, which is the variables. And we have in, where's my cursor gone? We need to specify now the parameters that are required in order to do that. And as you can see, this equation is down here shown y naught times exponential of minus ct. So we need to know y naught and c and t. These are the symbols symbolically that are there, and those that we want to convert to real numbers. So we need to specify that we need to know t and y naught, and we need to have c. Right, those three things that are in the solution, and that's sufficient because the other things are not really needed. So if you execute this code, you can see function handle, and it worked. We have three, in principle, accessible variable things, t, y naught, and c, and that function is y naught times the exponential of minus c times t. And you remember that notation for implicit functions, anonymous functions, the dot prior to the operator, meaning element-wise operation. Okay, so having our MATLAB function, we can go about and define something that we can plot. So y, if we want to have multiple plots, I don't know, we can try just our first function, or call it fun, or whatever you want to call that. And now we have to specify is equal to, what do you want to have as a variable? Well, obviously time. If we want to see a temporal profile, time needs to be our variable. And we take the algebraic function that we just defined, which is my. And then we need to specify the values for those three parameters that we have here. So time being a variable that's going to change, then we don't specify, just put the symbol here, but we put values for y naught and c. So we need to find them because I don't remember. C is supposed to be 0.1, and y naught we can use 10. Let's use this one here, y naught equal to 10. So we can do that by specifying y naught equal to 10 and c 0.1 in that order, right? In the same order as they appear here. So that would be our function. And then we know for every plot of a function, we need to define the limits, and we put them in parentheses from 0 to, I don't know what I need to look it up. It says 15. My script might be okay. Let's just try limits of 50, and then we can use f plot, function plot, of the thing that we have specified. So fy1 in the limits specified by limits, if that would just go away, in the limits specified by limits, and then we can see what happens. And we can see a plot immediately there. So obviously there's something that we need to change, and what we're going to change is that, I think, line width, and we want to have something thicker. So now we have a better plot. The background, why does it start from 10? Because we have specified 10 as the initial condition. That's all fine. What happens to that? So the variable y, right? If we would have to indicate that's now the solution of y, that is how the variable y behaves over time. And it starts from 10 and exponentially decays to a value that is close to 0. What do we see? Well, we can see exponential decay. It goes to 0, so eventually it will disappear. That's what it means, right? If things go according to this model that we have, minus cy, then y will start from 10, just decay and disappear, which also means that that will have an impact on the other variable as well. because y is not dependent on x. This is everything that we have. And the final thing that we might want to consider is half-life. Yes, we have calculated it before. You can do it, plug in the numbers here. But what we can see is that half-life is, of course, the initial concentration divided by 2, or the initial population divided by 2, so that's 5. So just visually, by visual inspection, if we just draw a horizontal line where it hits the function, it's about where I put this hand symbol, put it there. So that's about where we reach the half population, and the time point, if we then go down, will be somewhere here between, I don't know, around 6 or 7 time units. We haven't specified whether that's milliseconds or hours or years, but whatever model time is, that's how long it takes to go to decay to the half of the original population. Of course, one thing that we can do easily is now also go and plot the x function in a very similar way. So if I just do it in the same plot, oops, that didn't work, I make some space. If I just put it beyond this, I above it, and then I specify mx, the MATLAB function of s, and I need to get that from the solution for x. And then the vars are no longer these ones here, but the variables needed in the equation for x. I think I'll just have a look. Everything that is in this equation on here, right? So here's, well, let me make it a little bit nicer so we only see this. Everything that appears in this equation at the bottom now needs to be specified as a variable to be passed to the algebraic function. So t is definitely there, so we keep it like this. And then I will just copy that from somewhere. Then we have parameters b, x naught, b, comma, x naught, c, comma, g, comma, in the order of appearance in the equation, y naught as a last thing, and c we have already. So let me check it's correct. So we have t, then we have b, c, g, those three parameters, x naught and y naught as parameters that are passed over to the algebraic function, and which, if we want to now specify a function, I copy and paste this as well, and I specify a function x1 now, derived from the x variable, so taking the solution from the x algebraic function and specifying the parameters in the order in which they appear over here. So what I need to know is BCG and x naught, y naught. I go all the way up, where's BCG? B is 1, G is 1, C is 0.1, x naught 0.1, y naught 10. So we use those things, and I'll put that in here. So let's put it in as y 0.1, 0.1 if I have it here, then was it 2, and y naught was 10. So I need to count 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So in this case, it means that b is taken equal to 1 and x naught is 0.1. Let's just check those two. B is equal to 1 and x naught 0.1 and c 0.1 as we had it before. So that would be the definition of our function derived from solution to the variable x. And then if we want to plot both functions, of course, we have to specify hold on. Maybe you remember, hold on to keep both graphs at the same time. And then we want to plot this variable here, which is x1, so the first function derived from the algebraic x solution within the same limits, and we can use the same line width. But if we want to have a different color, I don't know, let's see how it appears. Okay, something is wrong here. I did some, I think the problem I specified mx from x, and I think I messed the 2 up. You see what I did? I changed the wrong things. This is the y variable, and this is the x variable. So I did that second thing, but I put it in the first place. So that's why the solution came out with you didn't specify the correct numbers of parameters for the correct function. So see what happens now. And the plot seems to work. I just take this error message away because I solved it. Just to make sure you see what I did wrong. I had specified the parameters for the y solution, but I had actually converted it to x. But x needs 6 rather than 3 specified. So this mixing up is now clarified. Now we have 6 parameters for the x function, and we're plotting the x function, and 3 for the y function, plotting the y function. And here's the corresponding graph. Okay, does it make sense? So what we can see is, I think I just put this thing in here. Yeah, I think that will be fine. So with those parameters specified there, blue is the solution for y, the temporal evolution of the variable y, the regulatory protein. Red is the temporal evolution of the X protein, so the protein of interest that we wanted to have synthesized. And what we see is under those conditions, never mind the exact values that are in here, from an initial very small concentration of X naught here near time 0, it rises sharply. And the reason is that this is modulated by the amount of Y present. So that is a regulatory protein in high presence, so values near 10 will make a quick expression, transcription, translation of that X protein, and therefore it will accumulate very quickly. But because the amount of Y decays exponentially at some point, that rise in protein X will be stopped, and below a certain value, so from 7 point something in Y onwards, it will actually start to decay as well, because overall protein Y goes to 0. Eventually X will also follow and decay to 0. So what we get is, in terms of X, we get a pulse of X caused by an initially high concentration of Y. Okay, so that's as much as I want to say, and that's how we worked it through with getting the solution of an equation using dsolve, and then converting it to a MATLAB function, just plotting it as a function of time, and we are able to see what the predictions are. Now, if we put in different numbers for those parameters, different initial conditions, of course, we can then study in detail the behavior of this model and, for example, compare it to an experimental situation, adapt the rise and decay, the half-life, and so on, in agreement with data. Good. Let's go to the final part. 30, good in time. Final part now is exercise 151. So here's a bit of a variation on the toggle switch being the content of the lesson. So you have a look at it. Toggle switch was one of the early approaches where it was said that synthetic biology is not just about synthesizing something, but synthesizing based on rational design. And rational design was interpreted as meaning I make a specific model of what I think going on, and then if I can realize that experimentally, I can have a comparison between experimental data, say in this case bacterial population, is that original toggle switch paper on. You have some link in the materials. And so the main key is here that you have a rational design, right? That these equations that are in the toggle system or in any model sort of represent a circuit or a way of interactions, how you imagine things to be, and making good assumptions. Of course, you can then do all the tools that we are doing here before then going into compare to the actual experimental result. And of course, this is a generic technique that we're teaching and that can apply to just about anything in the life sciences. And of course, you can also do, as you guessed it, to make models of the temporal evolution of the people infected with coronavirus and so on, make predictions about the next pandemic, when do you expect the next pandemic to happen. And of course, all the outcomes will depend on the assumptions that you put into the model. So the interpretations of the alpha here, is it just abstract parameters, or put in some numbers, that can be very political, and this is beyond our scope. So let's have a look at this task here, and it says use another system that has actually properties that are very, very similar to the toggle switch, but it's done mathematically in a different way. So rather than having a variable to the power of something, it takes the exponent of a variable, and that's what's happening here. Otherwise, it's structurally very similar, and equations are given here. So don't go into the detail of this, but if I remember correctly, this equation comes from the context of two neurons interacting. So it's like a mutual inhibition between two neurons that have contact with each other. It's like mutual inhibition. So I think that's where it originally comes from, meaning that, of course, when it comes to functionality of brain circuits or information processing in the brain in the context of neuronal circuits, you do exactly the same strategy. So it's in population dynamics, it's in synthetic bacterial population, and it's in neuronal system. Just about anywhere you can use this technique of modeling and studying models in order to understand what's going on, also to predict what's going to happen under certain perturbations. So let's have a look at this. Let's check this, this thing here. And what I have done is rather than doing it now by typing, just to save time, I have just put it in. And so you can get the same, you can get this from that script that I showed before. It's already in there. You can copy and paste that, make your task a little bit easier, and we don't need to type everything here. So this is a function definition, as we can see by the word function. So it needs to go at the bottom of the script at the very end. Then you can call this function from within that script. So we do y dot, like short, so just a name, but it's y dot for the derivative. It's what the function returns to the script when you call it. And the function has a name which is toggle sub dot, or toggle underscore dot is the modified toggle switch. So it's of course not a toggle switch, but it's something that works similar. So I just called it modified toggle switch. And then it has two variables. One is t, the time, which needs to go there by construction. That's the way we use the integration routine. And then all variables in this case are called y. So because this routine will sum up all the variables under one array, they go under one. So now it's no longer x and y with different names, but we'll have for the first variable y1, and second variable will be y2. Then the parameters, I don't really worry, just copied and pasted that from the assignment. Just say take alpha 1 equal to 10 and beta gamma equal to the values that you see here. So I just put it there. And then we need to specify some number, whatever the number is, you can work with that, but you need to specify because otherwise no solution will be obtained algebraically. And then I make an abbreviation for the left-hand side, du dv, which is of course nonsense if you look at y's, but you know that if it comes from the toggle switch was defined in terms of u and v. And then the equations. So we have the first variable, which is called y1, depends on itself. In which way? Negatively on itself. So it's a first order decay in kinetics, if you like, or it means if it gets very large, there will be a tendency to diminish by virtue of being there negatively with its own value. And then positively, it's affected by the second term, which is very complicated. If we just structurally switch to the second one, the second variable also depends on its own value in a negative way. So the derivative of y2 depends on minus y2. So it will be sort of capped. It will not go to very large numbers. It will be kept in small numbers. And then there's a positive term that looks structurally very similar to the first one. So let's see what that looks like. So that's a fraction, obviously. If I just take that apart here, you can see a fraction. On top is alpha 1 or alpha 2. Well, these are just numbers. Well, there's nothing to worry about. It's just 10. Right? Just put in 10 there and that's it. So nothing really to worry about. It's some number divided by what? The exponent of a variable times beta. So beta we know is a number, in this case 3, so nothing to worry about. The exponent of a variable. So what happens? You take the variable, but instead of putting in the variable directly, you take the exponent of a variable. So that means whatever that exponential function does, so it rises, right? Starting from 0, it rises, gets very large for large values of that variable, say y2. So what it means, if y2 has a large value, then the exponents will be very large. And because we're dividing by something, if that's very large, division by something very large means you have a very small number. So large amounts of y2 will make sure that the first variable, y1, doesn't have any positive term anymore. So what it means, it will tend to decay. And vice versa, if you have very large numbers of the first variable y1, then due to this being divided by the exponent of y1, you will make sure that the second variable will not have any or much of a positive contribution, but that this positive contribution will tend to 0. So this whole thing here will be tending to 0 because 10 divided by something very large will still be very, very small. So and then we just summarize the u dv and we call that y dot, and this is the output what we return. That's sort of the vector of all the derivatives, if you like, and that's how we set it up in order to be able to simulate it. Okay, what do we have? Modify the code. So we did that. Plot the concentration for certain initial conditions. And here it says U and V, but of course in our case, we remember this is Y1 and Y2. So let's define some initial conditions. So put that immediately. So let's do this first thing. First variable 0.1, second variable 1. So because we're collecting them in a single array, we can then specify the first variable will start at 0.1 and the second will start at, no, it's not 10, it's 1 or 1.0. So I just put those things here into something called y0, and I need to remember y0 are my initial conditions. Okay, that's it. So now I need to specify for how long do you want to simulate. So time span. That's what it's called in the script. So let's call it time span, and that goes in square brackets again. So we go from somewhere to somewhere. And what do we take? 0 to 15. It's what it suggests here, 0,15. Or we can modify that if we don't like it. Put a semicolon there because that's enough for us. So now you remember that time, comma, variables. So we have two things that we get out of the call of ODE. If we use that for, which one? 45, 23, okay, OD45, we use that for simulation, and we call it, and we get out of it the time variable and all the variables in one variable called variables. So then we need to specify which model do you want to have simulated. So we do that with the @ sign. And the function is still here, so I can just copy and paste that. It's toggle mod. And then we need the time span and the initial conditions. Time span and the initial conditions were called y0. So that's all that we need for simulation. And then we get the simulation results. Remember to suppress that output because it might be hundreds of numbers showing up if you don't suppress it. And then all that's left is a plot of that thing, right? So we need to have a plot. And the plot, of course, we want to see time versus the variables. Variables, LES. That's what we want to need, and we do refinement later. Okay, well, we'll see what happens. Let's just execute the code, and we can see that code runs and it shows nice graph. And maybe we don't need to simulate all the way. Maybe simulation up to 10 will be sufficient. So I do the time span from 0 to 10, and that's the graph that we get. So what do we get? We have two variables, and we can get this in. And if you remember, this was the first one. That was y1. And then we have y2 here. So the legend tells us blue is y1 and red is y2. In that order, what we see is from their respective initial conditions, they're taking different time courses. So they depend on each other. They're mutually dependent on each other, but they take different time courses. And the first variable, y, the blue one, stays close to 0, actually approaches 0. So very small values. And the other one, the red one, instead rises and then approaches a value that looks to be 10. I think it's not a coincidence. It has to do with a parameter that we set to 10, probably this alpha that we set to 10 initially. So we can find this diverging behavior, although symmetric, they are very symmetrically designed, right? Actually, it's the same thing in both equations, and alpha 1, alpha 2 is the same, beta, gamma is the same. So it's a perfectly symmetric equation, so that do exactly the same according to the mathematics, but numerically they behave very differently. Namely, they behave such that one stays close to 0 and the other one rises to a large value, which is 10 in this case. And so the idea of studying the system is, where does it come from? Why do they not behave symmetrically, although the equations are symmetric? And the suggestion then is, if we go back to that instruction, now repeat that simulation, but where does it say? Plot the concentration, use initial conditions that are 5 and 4 instead of 0.1 and 1. So do 5 and 4. Let's do 5 and 4. I think I just keep this here. So I make a copy of this. I comment the first thing, and then I put 5 and 4. Just so we can switch back and forth if needed. Put 5 and 4, 5.0 and 4.0 as it's according to the instructions. And then repeat that simulation. And of course we can do that, but we can also make sure that after the simulation, we can still see what happened originally by adding, before we go to the plot, hold on. You know this trick already, right? So we have the same as before, simulate over the same time span from different initial conditions now, but plot it in that same graph and don't erase the previous plot. Let's see what happens if we do that. And here's the result. So it works directly, and it now works in an interesting way, namely in which now we need to see which one is which. So the first variable of the second simulation is this one here. It's the yellow one. And the second variable in the second simulation is the purple one, or magenta, purple, let's call it purple. So it's not too complicated, so I'm not adding any other graphs, but just leave it like this so we can see what happens in this case. The first variable was the blue one. It started from 0.1. Initially, it stayed close to 0. What happens with the first variable now? Now we start from 5, from a high value here. And it also falls down as if wanting to approach 0 like before. So it starts from high, and then you remember minus y2, it says in the equation. So falls down, falls down, falls down. But at time point 2 here, incidentally at this time point 2, starts to make a turn and decides, well, actually, I don't want to go to 0, and then starts to rise again, and it ends up somewhere near 10. I think in the long run, it approaches 10 exactly. Right, so that's different. Now the other variable, second one was red, initially started from a small value and then it rose and reached 10. So it got there just growing, and sort of in an exponential approach to 10. Now it's the purple one. Now it starts at 4, so still a high value, much higher than before. Before it was 1, now it's 4. But instead of rising, it slows down, goes down, decreases, and I don't know if this looks like a change in slope here. That's a little bit sharp slope, low slope, but then it approaches 0 and stays there. And it means in the long run, this variable number 2 is now 0, whereas in the previous case it was 10. So the outcome of the 2 has swapped, right? 1 was previously 0. In the long run, 2 was high, and now they have swapped, although both initial concentrations are much higher. And this is our way to study the system, and I leave the rest for you. The solution to what does it depend on? Of course, if you look at the symmetry of the equation, there's a very simple answer to this. Whatever initial condition is higher, so which of the two variables is higher, will end up at the upper level of 10 under this case. So whatever the exact absolute values are, it only depends on which initial condition is larger, and that's the one that will end up at the higher value. So you can see here the red and the yellow. So if you just make sure one is larger than the other, you know where it will end up. You can actually predict without doing a simulation. Final comment. This is a case of bistability, and what it means now going to the phenotype, right? Now we have done some simulations, very abstract here, but if we go now to the phenotype, what it means. If that is a gene expression model, if it applies to a gene expression model, then what it means is that you will have different phenotypes, although the underlying equations means the same system as before. Just the preparation of the initial conditions, sort of the experimental preparation, depends which of the two phenotypes you express. And another thing that we don't do here, but that you can also do by perturbations, which means fixing the initial conditions by doing perturbations in the right way, in this case one larger than the other, you can swap the phenotype of a living cell under stable conditions. You can swap the phenotype with a single pulse if you know that they are in the situation where they are actually bistable. And bistable meaning you can have either one of the proteins highly expressed, the other down, or you have the other protein highly expressed and the other down. So two possibilities, two possible phenotypes, and that this is not just fiction, that this actually has been observed in bacterial cultures where people have been able to tune bacterial populations such that some of the bacterial cells went into one phenotype, and you do this with some fluorescence marker that you attach to one of the proteins here, and the other going into the other, and you could actually make them switch from one phenotype to the other phenotype in terms of expressed protein, and thus change on function that goes on downstream. Okay.